Welcome to Azad Academy. Today I will talk about neurotransmission. That means a communication system between two neurons or a neuron and effector organ. That means how a neuron can communicate with other neuron or other effector organ. So this is the communication system, you can say, in between two neuron and effector organ. So how it will communicate? Because nerves are generally are not directly connected. So it is a gap. So the main way of communication is chemical communication, the chemical messenger, that is the neurotransmitter. So through a neurotransmitter, the communication between two neurons. So I will just start briefly, and I will give you one thing, that is, I will explain in molecular level in all the events of the neurotransmission elaborately. But now briefly I will explain one after another all the events. Okay, so firstly today's topic is neurotransmission. Okay, so you can elaborate it. You can just fraction what is neuron, or you can say neurotransmitter, or this is the mission. So this is the multiple events, like that. Okay, so firstly you have to know the neurotransmitter. What does it mean? Neurotransmitter, is a chemical messenger. You can think hormone is also a chemical messenger. It can communicate throughout the body, release from gland and just travel around throughout the body. So what is the difference between neurotransmitter and hormone? I will not go for that, but I will just make you clear. Neurotransmitter is also a chemical messenger, and it will just communicate between two neuron or effector organ. That's it. But is it enough? No. The main criteria to be a neurotransmitter is it a chemical substance that must be released from the neuron, must be released, and it would be very better if it will synthesized within the neuron and it will release from the neuron. That is the true neurotransmitter, not pseudo. Some neurotransmitter can synthesize from the outside of the neuron and it can store within the neuron. But mostly you can say two criteria you can consider, but one criteria you must follow, that is it must be released from the neuron. If you think this is the neuron non-terminal, so from here it will release. And another criteria, it would be very good for it if the neurotransmitter is synthesized within the neuron. So it will synthesize within the neuron and it will release within the neuron. That is the true neurotransmitter. So another third criteria should be must be followed by a chemical substance, that is it must carry message. Okay, so this is the chemical messenger. That means neurotransmitter is a chemical substance that will release from the neuron and it will carry a message, because neuron can release some other substances. You can say neuropeptides, multiple number of neuropeptides can be synthesized from the neuron. So these are not neurotransmitter in a true sense. So neurotransmitter means chemical substance that will be released from the neuron and it will give message to another neuron or another effector organ. So that is the neurotransmitter. Okay, so all way through the neurotransmission system, I will explain through multiple events related to neurotransmitter. So what would be the events connected to the neurotransmitter? First question may come in your mind, that is one thing, first of all, synthesis. Synthesis. That means how it will synthesize, or second question may come, where it will synthesize. Okay, already I said it will say mostly it will synthesize within the neuron. So where within the neuron? Neuron has several parts. It has dendrite, it has soma, it has axon. So where? exactly it will be synthesized. So you can think like this is the nerve terminal neuron, here is the dendrite, you can say this is the soma, this is axon. Okay, so where the neurotransmitter will synthesize. So synthesis of neurotransmitter. So question, before going to the site of the synthesis, I will explain it in two categories. One is firstly here the types of neurotransmitter. Types of neurotransmitter. What types of neurotransmitter? Actually, I will not go for the chemical classification, the chemical precursor. I will not go for that. Actually, I will go for the molecular size classification, molecular weight. So it is basically two types. One is uh, a large neurotransmitter, or you can say neuropeptides. Yeah. Not both are different things, but neuropeptides is not neurotransmitter, but it can modulate the function of the nerve function. And second one is a small neurotransmitter according to the molecular weight. So why I will discuss about it during the synthesis discussion part, because the large neurotransmitter or neuropeptides are basically, these are protein in nature. Okay, protein in nature. So all the machineries to produce protein are present within the soma. That means soma has nucleus, the blueprint of the protein. It has ribosome, the machine of the factory of the protein. It has endoplasmic reticulum, is the modulator. It has Golgi apparatus, that is the processor. So all the machineries to produce a protein or peptide, those are present in soma. So large neurotransmitter or neuropeptides are synthesized within the soma. Okay, so here. So I will just categorize it in two things: large neurotransmitter. So I already said neurotransmitter. It must carry the message. So protein in nature, and it will act as a neurotransmitter. So the example you can think like vasopressin. You can think about oxytocin. You can think corticotropin releasing hormone. These are the neurotransmitter, which are basically protein in nature, and of course those are synthesized within the soma. Okay, and some large, of course large as is the peptide or protein, so assembling form of amino acid, more number of amino acids, so large neuropeptides. So neuropeptides. So example of the neuropeptides, you know, the substance P, you can say neuropeptide, or you can say like you can say endorphin, you can say dynorphin, you can also say cholecystokinin, that is CCK. So all the neuropeptides. So neuropeptides, multiple numbers of neuropeptides are there. So that can be neuropeptides or neurotransmitter, protein neurotransmitter. Large substances will be produced within the soma. Okay, so this is the site of the synthesis of large neurotransmitter, protein in nature neurotransmitter within the soma, and of course that will travel down to the terminal, and it will just retain within the synaptic vesicle of the terminal, and then finally it will release. Okay, so the site of the synthesis is of course the soma, and of course you know the classical way of transmission is the terminal. Although some neurotransmitter can release from the dendrite, the dendritic transmission, that is the different things I will discuss in separate video. So this is one, and the second one is a small neurotransmitter. So you can consider neurotransmitter, a small neurotransmitter. So molecular size is narrow, and it will work very fast, very quick. And if I just categorize in different way, that is simple amino acid. You can think like glutamate. You can think about GABA. You can think glycine, like that. These are the simple amino acid. And second one is biogenic neurotransmitter, is small. So this is just one step or two step down amino acid to neurotransmitter. Like you can think about serotonin, you can think histamine. Okay, and some multiple events conversion of amino acid to the neurotransmitter, like you can say noradrenaline, you can say dopamine, noradrenaline, like that. And so these are the biogenic. And also some small neurotransmitter are gases, like nitric oxide, carbon monoxide, like that. Some neurotransmitter itself is a class, like acetylcholine. Okay, so these are the small size neurotransmitter. So what would be the site of the synthesis of the neurotransmitter? So synthesis of the neurotransmitter would be both in soma and nerve terminal. Okay, so in axon within the nerve terminal, that is cytoplasm, it has the required elements for the simple conversion of the amino acid to neurotransmitter, or some multiple enzymes within the cytoplasm of the terminal. So the small neurotransmitters is mostly or mainly synthesized within the nerve terminal. Okay, I repeat it, mainly or mostly, not I am saying like only. So mostly or mainly synthesized within the nerve terminal, because this conversion is very quick and it will store within the vesicle and then quickly it will release, and mostly it will synthesize within the nerve terminal. Okay, but also these neurotransmitters are synthesized within the soma. That means I am trying to say soma can also produce, also synthesize a small neurotransmitter, and nerve terminal also. But mostly the synthesis is happening within the nerve terminal. But the small neurotransmitter can also synthesize within the soma. Okay, so when soma is the synthesis site of the small neurotransmitter, it will travel down or trafficking from the soma to nerve terminal, and then it will store within the synaptic vesicle in the nerve terminal. Okay, but some neurotransmitter are just released, synthesized and released at the site of the synthesis. Like you can say nitric oxide, it will synthesize from here, it will release from here, it will synthesize here, and it will release from here. Histamine, it will synthesize here and it will release. But you can say GABA, you can say dopamine, you can say your noradrenaline, that can be synthesized both in soma and also nerve terminal, but mostly nerve terminal, but when it will synthesize within the soma, it will travel down and then finally store in the synaptic vesicle for further release. Okay, and already I said, slightly I said, some small neurotransmitter can release through the dendrite for the special purpose that I will explain in separate video. Okay, so this is all about the synthesis of neurotransmitter. So actually this is the class synthesis that can be synthesized from the precursor molecule. Already I said that amino acid. This is the conversion of amino acid, and some are acetyl CoA and then choline, then acetyl CoA, choline. You can say the vasopressin from protein, oxytocin protein, that is the come from basically from the blueprint of DNA signal, then messenger RNA, then protein, and then finally processing, and it will produce vasopressin and oxytocin. So this is the synthesis process or site of the synthesis of neurotransmitter. Okay, so this is the first event or first step of neurotransmission. So now I will go for the second one, that is storage. Second one is storage. So how or where it will store? That is the question. So already I said in generally, commonly neurotransmitter release from the nerve terminal, that is from your axon, and axodendritic synapse is very common. So if you consider this is the nerve terminal, and here would be a vesicle in which neurotransmitter will retain. Okay, so storage means, again I have to go in two categories because already I said large and small. So of course vesicle would be in two different, because size or molecular size is different. So vesicle of course would be different. The question is, vesicle, where vesicle will be produced from? Also vesicle is protein in nature, so the vesicle also will come from the soma. That means in soma, in endoplasmic reticulum, then Golgi apparatus, vesicle will produce, and that vesicle will travel down or trafficking from the soma to the nerve terminal. And where in the nerve terminal, and what are the types of vesicle? Now I will explain now. So come to the point. So storage means the neurotransmitter will store where? In vesicle. Vesicle is a kind of sac that will retain the neurotransmitter. So this is the vesicle. So what are the two types of vesicle? Just now I will say large vesicle name is large dense core vesicle. Large dense core vesicle. This vesicle is for neuropeptide, or you can say neurotransmitter, or type of protein in nature. So protein in nature, neurotransmitter or neuropeptides that will store within the large dense core vesicle, that will be present within the nerve terminal, and that will retain the neurotransmitter. That is, you can say vasopressin, oxytocin, you can say the cholecystokinin or substance P, neuropeptide Y, or any other neurotransmitter with protein in nature. Okay. And second one, that is small clear core vesicle or you can say small synaptic vesicle. So this is for your small neurotransmitter. Already I said that is directly amino acid, glutamate, GABA, glycine, like that, or you can say histamine, serotonin, dopamine, noradrenaline, acetylcholine. These are. So this is the vesicle. So two types of vesicle. Okay. So in generally, one neuron, one neurotransmitter. This is very common for your CNS or even for your PNS. A single type of neuron will carry a single type of neurotransmitter. Like dopaminergic neuron will carry dopamine neurotransmitter. GABAergic neuron will carry GABA. You can say your parasympathetic nerve, that is cholinergic neuron, will carry acetylcholine. So single neuron, single neurotransmitter, and single neuron, single neurotransmitter, single type of vesicle, that is very common. Okay, but co-release, co-transmission, or co-packaging, or you can say multiple or dual neurotransmitter can be present within a same nerve terminal, same neuron. You can say a nerve terminal can carry a multiple number of vesicle, of course, that would be same or different. Okay, so I will make you clear by some example. Like, suppose this is your nerve terminal. Mostly this is very common. This is neurotransmitter vesicle, and these are carrying a same neurotransmitter. If you consider these are the dopamine, so this is the dopaminergic nerve. This is very common all over the body. This is classical and this is mostly happening. Okay, and now I'm coming here. If you think this is the nerve ending, and that is just carrying a large neurotransmitter vesicle. Here is large neurotransmitter vesicle. You can think, I will explain in different example, or just here I can give you another things. This is a small neurotransmitter vesicle. So what will happen? A large neurotransmitter vesicle will some transporter protein will be embedded on the membrane. So through this transporter protein, neurotransmitter will enter. If you think this is vasopressin, so this is the vasopressin transporter channel. Okay, such transporter protein. So that will store here. This is on type. Here another type of neurotransmitter. Suppose you can think oxytocin. So this is, you can think vasopressin. You can think this is oxytocin. So these are oxytocin. So separate vesicle, but present within a single neuron. Okay, I will not add here the small one. I will explain in separate example. So large vesicle, large dense core vesicle, that is just carrying here by a independent packaging pathway. Independent packaging means independently individual transporter protein, it will work by some mechanism. Already you know, here proton pump 80-page pathway. I will explain in separate video, already I said, but briefly I explaining now. So a single nerve can carry dual or multiple separate vesicle that will carry a separate neurotransmitter. You can say oxytocin and vasopressin. Also, it is possible like that. That means one vesicle, it can carry both oxytocin and you can say vasopressin within a single vesicle. Okay, it's also possible, but very less common. But mostly, already I said, most common one neuron, one neurotransmitter. This is mostly common. But separate vesicle, separate two individual vesicle is also possible. But within a vesicle, two neurotransmitter is also possible. Here, vasopressin and also corresponding vasopressin neuropeptide also can be present within the vesicle. Okay, so this is the large example is single vesicle will carry a large vesicle through the transporter, single to single. This is example, and this is individual but separate, and this is a single vesicle but dual neurotransmitter present. So this is the storage way of large vesicle. This is possible. Now I will say with the same manner, like this is a small. This is the small neurotransmitter vesicle, and it will store a small neurotransmitter. Already I said dopamine, so dopaminergic, GABA, so GABAminergic, acetylcholine, so cholinergic neuron. This is mostly common. You can mark as a star mark like that. This is common. And here another things, that means a small neurotransmitter vesicle, both are different vesicle, and here you can add some transporter. I will not showing here the proton pump ATPase pathway. So this is the only storage condition. So here you can say here GABA, and you can say here is glutamate can be present within a single neuron. In two separate vesicle can carry two different neurotransmitter at a time. So it is also possible. Suppose if you think within your brain, limbic system, that is possible, hypothalamus, this is possible. So it is possible, such type of things. So this is one type of matter. Another thing is one synaptic vesicle, and this is a simple single neurotransmitter here, a small neurotransmitter, and this is a large neurotransmitter vesicle, large. It is also possible. That means a small neurotransmitter vesicle, it will carry a small neurotransmitter, and it will also simultaneously can release a neuropeptide that will modulate the function. Okay, this is very common. Okay, so co-release or co-packaging. So this is co-packaging. That means a single vesicle, the separate neurotransmitter transporter packaging. So this is the co-packaging. So independent packaging already I said, and this is co-packaging. Packaging. Okay. So here, so individual separate vesicle, both are carrying a small neurotransmitter. Here one is small neurotransmitter and one is large neurotransmitter, and mostly single neurotransmitter, single neuron. So these are the small types. And you can say, is it possible within a same neurotransmitter vesicle, small neurotransmitter vesicle will carry at a time uh, separate transporter, sorry, it is also possible. Like you can think here, this is your dopamine and this is your GABA, sorry, or glutamate, dopamine or glutamate, like that. So this is the co-packaging. The same vesicle are embedded with separate different transporter protein, and it is carrying a different neurotransmitter. That is very uncommon, but very possible within your brain as well as your rest of the body, CNS or PNS. But mostly such type of neuron are present within the brain, because brain always work in diversified way. Okay, so these are the storage way. Okay, so storage, the separate large and small vesicle, and that can carry in different way, in different situation, different neuron. And always I am trying to say that is one neurotransmitter is very common, but nowadays it is not like exception, it is also usual. It is very possible within your brain and your PNS, CNS and PNS. Not exception like that, not a single occurrence or very few number of neuronal like that, very common. This can be, when it will release, then it can be said co-release or co-transmission. This is common phenomenon nowadays, but still now this is very common. This is mostly happening. Single neuron, single neurotransmitter. Okay, so this is the storage way. Now I will go for the third event, that is your release. Third is release of neurotransmitter. So already I said within your nerve terminal, neurotransmitter vesicle are there, and that vesicle will release the neurotransmitter. How? So when I will say release, so in which pattern the vesicle will be within the nerve terminal? That is first come, first serve, like that, or how? So if I just try to explain it, then I will give you 3 examples of the situation of that neurotransmitter vesicle within the nerve terminal. That is, one is ready to release, or you can say ready pull, and second one is, you can say reserve, or you can say cytoplasmic pull, or third one you can say recycling, or you can say recovery pull. That means, here I will explain. This is a neurotransmitter nerve terminal. and here one zone is there, that is active zone. Active zone. Why it is called active zone? Because it has a multiple number of protein. I will explain in elaborate way in separate video, but I will just now give you some brief knowledge on it. Some protein like snare protein, like you can say stagmin, you can say snare 25, you can say monk protein, like different proteins are there. And also the special criteria of that active zone is here it carry a multiple number of calcium channel, multiple number of calcium channel, that is calcium channel. Okay, so this is the active zone criteria area. And here, so ready pool means the neurotransmitter or neurotransmitter vesicles are ready to release the neurotransmitter. So where they will stay? They will stay just very adjacent to the active zone, and as a docking state, priming state. Okay, it will just wait for signal, stimulation, action potential, like that. So how it will stay there? So the vesicle, actually I'm just drawing here. Vesicle will carry a multiple number of protein also, different types of protein. I will explain in separate video already I said. I'm just giving you some example like synaptobravin, synaptotagmin, you can say RAB protein. Multiple number of protein are there on the surface of the synaptic vesicle, and that protein will interact with the protein of the active zone, and it will just make a complex, and finally it will help to release. How? So firstly, that protein, and here some protein, okay, some RAB protein, and here monk protein or RAB effector protein will attach each other, and then it will here it will work monk protein and stagmin protein, snap 25, and then interact. I will explain a separate video again and again. I'm trying to say. So now, then what will happen? That ready to pull vesicle will be there. So that pull will be there here, and here just interacting with the proteins. Okay. So this is, this is first step is docking. Docking means just protein attachment. It is very close to the active zone. Okay, and what will happen then? Then it will just find out the active place of time to release. That is the priming. Some protein complex will form, and it will just translocate, and it will find a specific site of the active zone to release. So that is the priming. Okay, this docking and priming is the prior events of the action potential. That means that will happen before the action potential reach to the terminal. Okay, so without action potential, neurotransmitter release is not possible. So if you want to know more about action potential, you must watch my video in my channel. So you can go for action potential. That will be very helpful for to understand these events. Okay, so what will happen when a neuron will stimulated, excited? Then what will happen? The action potential will truly generate in the halo, and then it will travel down here, and then it will reach to the that way. Okay, so this is, you can say, this is the sodium channel, and that will enter, and it will cause depolarization, and by the influence of depolarization current, calcium channel will open, and then calcium will bind with enter into the nerve terminal, and it will bind with the synaptic on the surface of the vesicle, and then it will change the conformation, and it will cause the fusion of this membrane, the nerve terminal membrane and the vesicle membrane fuse. So that will be the fusion. Third stage is the fusion. Okay, so fusion means actually this is it will fuse the two membrane and it will release the neurotransmitter. So if you if you think this is the neurotransmitter, these are the neurotransmitter, so it will fuse and release the neurotransmitter. So this is the release process. Okay, so how the fusion will work and what type of fusion is possible. It is partial fusion, that is transient fusion, or you can say there is a kiss and run theory, or also you can say the full fusion. So what type of fusion is possible and how fusion is possible, I will explain in separate video. This is the fusion. Okay, so overall after fusion this process is called exocytosis. Okay, so when fusion will happen, when run and kiss and run, that quickly that vesicle will again come out from here and it will repack the neurotransmitter and be ready to release again. And when the full fusion happening, then the endocytosis process will be here, there. Endocytosis process, the Catherine protein will be attached, and then finally the vesicle will form again. Vesicle, not reload. After that it will reload. Okay, that means I missed one thing. Vesicle will be there, and neurotransmitter will reload through the transporter within this area. So this is the maturation of this vesicle. So when the recycling will happen, then again it will it will take part in the loading process of neurotransmitter, and then it will again take part in the release process. So this is the release of the neurotransmitter. Simply I'll say docking, priming and fusion, and finally the name is exocytosis. And basically here the action potential depolarization current will play an important role for final fusion or final diffusion of the vesicle and the membrane. Okay, so completely on molecular level, what type of proteins are there and how they are interact with each other and how they will make the conformational change and open up the membrane, I will explain in separate video. Okay, so stay with me. Okay, so this is the third stage or third event of the neurotransmission. Now I will go for the very quickly. I will go for the fourth stage, or you can say fourth event. That is the receptor binding, or you can say biological activity. Okay, that means already I said a neurotransmitter will release from the synaptic vesicle from the nerve terminal, and it will just come out here and it will stay within the synaptic cleft, and here is another neuron, that is the presynaptic terminal, and this is a post synaptic terminal. Post. Okay, so here the corresponding receptor will be there. So if this is the neurotransmitter, these are the neurotransmitter, and it will be within the receptor. That means the neurotransmitter will bind with the corresponding receptor. And what does it mean receptor? The receive, that will receive the neurotransmitter and change its conformation, and it will induce its signal transduction process, and it will make activation of this neurotransmitter. Or if here is the effector organ, if you think this is the effector organ like heart, so here would be the receptor. So if you think this receptor, this neurotransmitter is acetylcholine, so this can be the M2 protein, M2 receptor, and here neuron, it can NN receptor, like that. If you think this is the these are the noradrenaline, so that would be the receptor of alpha or beta. Here it will be the beta 1 or beta 2 receptor, like that. Okay, so receptor binding means after releasing of the neurotransmitter, it will stay within the synaptic vesicle, synaptic cleft, and then it will just search the corresponding receptor and it will bind with the receptor. And then through the binding of the receptor, it will activate the corresponding neuron or effector organ. So this is actually the main goal of a neurotransmitter or main goal of the neurotransmission. So this is the biological activity. Okay, so this is, so I will not go for the details about the structure of this receptor and how it will change the conformation, what types of the receptor, this is G protein coupled receptor or ion channel receptor. If you want to know more about it, then you can go for my another video, this is receptors, types of receptors. Okay, you will get all the information over there. Okay, so now I will go for the fifth stage, or you can say the last stage. Okay, so that means the fifth stage is the degradation or elimination, or you can say fate of the neurotransmitter. Fate of the neurotransmitter, or you can say degradation, or you can say finally elimination. Okay, so what will be the main things? Main things is, again, this is the synaptic terminal, not terminal, and this is your postsynaptic part, and this is your synaptic cleft, and you can so say these are the neurotransmitter. So what will happen there? What will be the fate of the neurotransmitter? So some neurotransmitter will bind with the receptor and it will activate the receptor and it will give the biological function. Already I covered that part. Some neurotransmitter can be degraded through some enzymes which are basically present there. Enzyme. You can say, if you think about acetylcholine, same acetylcholinesterase enzyme there to degrade the acetylcholine. If you think about adrenaline or noradrenaline or dopamine, then catecholamine O methyltransferase enzyme are there. So some enzymes will degrade the neurotransmitter. When they will degrade? Before binding to the receptor or after completion of the work. Okay, but our main desire is to the degradation of after completion of the activity. But sometimes when excessive amount of neurotransmitter will release, for the fine tuning or fine balance, some enzyme will degrade the excessive neurotransmitter. Okay, so enzymes are there that will degrade the corresponding neurotransmitter. Some neurotransmitter can degrade within the presynaptic part, like monoamino oxidase enzyme are there that will degrade the neurotransmitter. So degradation is one of the factor before and after the activity. Another is that you can say reuptake, or you can say recycling. Both are connected, both are very similar word but has some core difference. So reuptake means some neurotransmitter can go back to the presynaptic terminal through the reuptaking channel. Suppose if you think the serotonin, so serotonin reuptake receptor are there. So this way they will again come back. So reuptake. You can consider as a recycling, but sometimes if you think about acetylcholine here, so if the acetylcholinesterase enzyme degraded, then it will convert into acetyl, and this is the choline. So acetyl will again degrade by the blood, and choline will come back here through this channel, sodium and choline exchange. So choline can be re-enter. So this is the recycling. Okay, some neurotransmitter can recycle. Also, some neurotransmitter can reuptake by some channel, and recycling can also be possible by glutamate, glycine, like some neurotransmitter. So some neurotransmitter can reuptake or recycle. Okay, so recycling actually does not mean actively it will enter. Reuptaking is the complete matter, but recycling, if you consider as like acetylcholine, then not acetylcholine will re-enter, the choline part will enter into the nerve terminal. Okay, and then another thing, some neurotransmitter can diffuse into the glial cell. You know the glial cell is the helper cell of the nerve, protector cell of the nerve, that will diffuse to that area. That is reuptake, you can say like that. And also it can diffuse into the extracellular area, extracellular part. Also you can say it will diffuse into the bloodstream. Okay, so degradation or elimination or reuptaking or all over the fate of the neurotransmitter is that. Fate means what would be the life event of that neurotransmitter. That means it can bind with the corresponding receptor and it can work, or it can come back again, that means reuptake, or it can degrade, or it can just lost its destination. It can go into the bloodstream, or it can go to the extracellular space, or it can go to the another cell, taken up by the another cell. So this is the fifth phase, that is the degradation, you can say, or mostly you can say elimination, but commonly you can say the fate of the neurotransmitter. Okay, so this is all about the fifth events of the neurotransmission, or completely I will say this is the process of neurotransmission. Okay, so I am trying to just explain here briefly or very roughly all the events of this neurotransmission process, and I will make the lecture every step in more elaborate way as a molecular level. So till then, stay with me. Thank you very much.